Meet me in the nucleus. It's going down. Meet me in the cytoplasm. It's going down. Anytime your cells meet, it's going down. Yes, it is going down today. Not only do we have another video coming back at you, but today we are talking about protein synthesis. Now, in the interest of time, this video is going to be broken down into two separate videos because I don't want you to sit in front of a screen for 17 minutes all at once. I know sometimes it's too much of me to watch in 17 minutes. You need to break it down and take a break, come back and watch it another video. So that's what we're going to do. This first video is going to go by very quick. It's going to be kind of a review of what we learned in our previous lesson, and then we're going to get into transcription. The next video, we'll talk about translation and do some practice work with that. Are you ready to take this ride, this protein synthesis ride? All right, so here we go. Our first stop, again, just taking a visual look at protein synthesis. It is two parts, transcription and translation. The first step being transcription. If we go back to look at our notes on comparing RNA and DNA, we notice one or two distinct things. First off, DNA showing that ever so popular double helix structure, and then RNA is a single strand. Just some more review notes for you. Remember our base pairing rules when it comes to DNA and RNA. There is no thymine shown here in the table. There is no thymine in RNA. That now becomes replaced with uracil. So uracil replaces, oh, my pen's not working. Uracil replaces thymine. Okay. That's what we're stuck with, right? Uracil in RNA. And RNA is that single strand, is not a usual, is not a lovely double strand like you get in DNA. And recapping our three types of RNA, we have messenger RNA, we have rRNA or ribosomal RNA, and then we have transfer transfer RNA. These are very easy to remember. M for messenger, R for ribosomal, and then T for transfer. Transfer being the last step, it's bringing over the amino acids to the ribosomes. That's the best way to look at this. And then ultimately, what protein synthesis is showing here. DNA holds what we call the genetic code. So everything that makes each one of us unique and special, you can find in DNA. And why in the world is my pen not working? We're going to have to do this with the mouse. So each gene is coded with a different set of proteins. This segment of code we call, again, these are the genes, the traits. So blue eyes, pointed ears, big nose, all that is proteins that are coded together and gives us our genes or our traits. Now let's talk about transcription. There's two steps to protein synthesis. The first is transcription. And when I learned this, the easiest way for me to understand the order, I thought about the word transcription, right? Breaking the word down. It has a word script in it. I love movies. I just saw Black Panther 2. My mind is blown. If you haven't seen it, go see it. It's an amazing movie, right? But with all movies, you don't get the finished product until you have a script. So script comes first. That's how I remember transcription. We're taking the idea or the DNA, and now we are creating mRNA. So you're taking a thought, an idea you had, maybe a writer had a great idea. Now they're putting it on paper and they're turning it into a script. Once you get that script, then the actors and the actresses and the directors, they take that script and they turn it into a movie. Or for us, we take mRNA and we turn it into a protein. So, where does it all happen at? Transcription happens inside the nucleus. This is where we take the DNA and we turn it into, or we create a mRNA strand. Now, everybody should not be like, this should not be the first time you see the word polymerase, because we talked about it in unit 4. This time we have RNA polymerase that creates the complementary bases to the DNA. As you can see from this picture here, we have our DNA strand here at the bottom, and then RNA polymerase comes down and lays down the complementary bases. And as a quick reminder for you, I have down here at the bottom our base pairing rules. Remember, A goes with U and G goes with C. Now, to see who's been really paying attention, we have a little practice for you. I want you to take about 30 seconds. Oh, and never mind, because there goes the answer for you. I was going to give you 30 seconds to match it, but here's that. I always remember anytime we're doing anytime we are matching our mRNA to DNA, make sure we always have our 5' and 3' at the end. As you can see here, A with U. Anytime you see a T, I'm sorry, I put A to A. Anytime you see a T, you know in DNA or in RNA, there's going to be U there because we don't have any thymine in RNA. Let's clear that off, and I think that's pretty much it for transcription. Not a whole lot to go on for transcription. Make sure you go back or you come back to the channel, watch the second video on translation. We're going to walk through the second and final step in protein synthesis. See you later. And remember, I have no favorite classes because I enjoy all my classes equally. That's true. All right. See you next time. Peace.